so we do want to talk about methods and results. This is the section that you should be looking at, and this is the footnote that you should be looking at. They have the program right here, where they have two cysteines, I plus j plus k equals 18. But realize that I, j, and k can vary. And so they let it vary, and yet they did it twice, and both times they came up with the same kind of spacing for the cysteines. So the fact that the cysteines are spaced normally is significant. Right here, actually, they give a little more detail. J could be anything from 4 to 10. And yet, what do you, let's count up the J's. Let's count up what J is between C and C. So let's count 1, 2, 3, 4, 5, 6, 7, 8, 9, 5, 6, 7, 8, 9. So it could have been anything between 4 and 10, and yet it ended up being 9 in both cases. That's significant. The other thing to look for is, was it truly random? They had an N and S codon. You could actually look up. It was probably biased a little bit because you can't be truly random, but you make random nucleotides, and they will end up giving you random amino acids. It could be interesting if you wanted to geek out on it to see what an N and S codon. By the way, N and S represent multiple nucleotides at once. N can be any of the 4. S is actually more limited, and they chose that deliberately. You can look at that to see if that's the case, if that would have biased the pool at all. But they're not. It doesn't really matter if it's completely random. It just needs to be random enough, right? Because your real question is, where does it bind? So I already described the bacterial phage display. The important thing is it's actually a virus that infects bacteria. And so it has its own, I said DNA, but I wasn't quite sure. It might be RNA virus. Whatever it is, it infects bacteria. So that means that you can elute it, amplify it, infect new bacteria, and make more of it. And you can even do some different panning cycles to enrich. The ones that bind best will bind more. And so the more you do this, you will get more different things. I don't believe the amplification will re-randomize, though. I think you have a random pool at the beginning, and you're simply selecting from it. But it's things like that. How does it work? And that's what you'll want to get to explaining. Most of your stuff, phage display is a random sort of design technique. Your design techniques will be a little more rational, okay? And they're going to be tricky. There's going to be parts of it that you won't be able to explain or understand. That's okay. You want to find the key idea. Like in this case, the key idea was it's a virus, so it has a protein coat that expresses the protein in the DNA. And once you get the DNA out, you can sequence the DNA. You can amplify the DNA, and you can sequence that up, and you can tell exactly what was enriched. So this is where the results are. So hopefully, and I also showed this at the beginning. So hopefully you saw not just the spacing of the cysteines, but you see both times the GELVW sequence. Be interesting to look at. I don't believe there's any matches that you get in any other part of it, but it'd be interesting to double check their work to see if you see something that's kind of a minor match. I don't see anything though. And the reason why they were sticking to 20 residue peptides is because they were doing all these not by protein expression, but by protein peptide synthesis. They were found to, basically they were synthesized, and so I just want to say when you look at that footnote, guess what you see? 9 FMOC, FMOC chemistry, which is exactly what we talked about way back in Biochemistry 1. We talked about how FMOC was the chemistry that you use for this. And you can even see how they purified it, reverse phase chromatography. But that detail is not important enough to fit into a 10-minute talk. The other thing that they did is they used ELISA. This is another thing from Biochem, and it's also from other classes, to detect binding. The basic thing about ELISA is you run it in 96 well plates, so you can run 96 experiments at once. Less than that because each of them is a different concentration, but you can run a bunch of experiments at once. And you can look at the concentrations that they used. They were able to use concentrations down to nanomolar. So that's pretty low concentrations. And they basically, the nice thing about ELISA is it turns color when it binds. And so you can look at that, Well, I don't have a picture of it because we talked about it briefly in biochem. But if you're a little foggy on that, this is where you go back and you look it up and you can look up examples of it. The important thing to me is actually you're going to be doing binding assays, and the concentrations of the protein that you use in the antibody binding assays are going to be very important. And so look for those. This is not the last time you will see micromolar, nanomolar units because it's going to be our research. So remember that they had the two peptides. Right here they had, they did libraries, they did it twice and they got the same core. And so they made a peptide out of the core sequence and they cut off the end, so it's only a 13 residue core. Incredibly small. And yet it binds, it inhibits binding with a 25 nanomolar equilibrium constant. It's pretty low. The nanomolar binding is very good, especially for something that small. So even though it's short, it binds 200 times more rapid, more tightly. And it's actually 25 nanomolar compared to protein A and protein G, which are 4 times larger, and they bind around 10 nanomolar, which is only about twice stronger. So this is like, I don't know what the animal is that's like efficient. Is it like the shark that is supposed to be so optimized for its job? The shark is a predator that's optimized. This is the binding peptide that's like the shark. It's been optimized to extremes. So we will be dealing with these kind of numbers later. If you want to look at the crystallography, you definitely can. The one thing to look at for that is what is the resolution. And you see here the resolution is 2.7. If you remember, a good resolution was around 2. 2.7, it might not be the sharpest resolution that you can get, especially these days. And that is why we have the thing that I pointed out about how you can't see you can't see the hydrogen bonds for sure, because the residues could be a little bit off this way or that way, and hydrogen bonds are so dependent on direction, it really matters. But they got the crystal structure, and the thing they need to see is where on this big thing is it located, and there it is on the blue. So there it is at the same site. The other thing that you might notice looking at this, there's actually 2 sides to the antibody. The antibody FC region is symmetric. They don't usually show the other side because they don't need to, it's exactly the same. But they do show it in this figure, just so that you can remind yourself, oh yeah, they're seeing what you expect to see. But a lot of times if you have a symmetric situation, you just don't show it all. And so here's the crystal structure, and we want to look at that, the GELW sequence. You see there's the W sequence. It has a turn down here. Here's the 2 cysteines. And there's a glycine somewhere around here that's probably helping it turn. So you can connect the sequence with the structures here. And then they have several figures where they try to help you. And so you see which ones, you can see which ones I chose to illustrate. I could have chosen this one as well. This is one where they basically colored the binding site. You see there's the binding site there. And they colored it according to how many proteins does that site bind. So you can see that blue means it binds only 1 protein contacts that part of the 4 different, I'm sorry, it's only 3 different things here. They're comparing, they're comparing 4, sorry. It's the FC site, protein A, G, and rheumatoid factor. They had to throw out the FC receptor complex because it was 4.5 angstrom resolution. That's just low. You can't do anything with that. So they didn't even want to mess with it. But basically, if you had only one of those contacting the protein, you color it blue. If all of them contact the protein, you color it red. And you can see it makes like a target where the 3 other things, they are centered around that site. Where does the FC bind? The FC protein bind? Yeah, they call it FC3. FC3 binds where the green is. So it's like you dipped it in paint and stuck it onto the thing. And you see it's like it's stuck to the target. It's definitely binding the same place. You definitely can't get away from that fact. So this is where they look at the topology. It has the groove and it has these things on either side. And these are the 4 different, the 4 other things. There's protein A, protein G are some of these, and then they have the rheumatoid factor. And they're just totally different. But the thing is, how does the green part change? How does the FC region change? Is there conformational flexibility? And that's something that we keyed into on the very part of class. And there is some, but it's not backbone flexibility, it's residue flexibility. And that's what I want you to see right here. This is where they had the movie. It's low res, but that's what we could do. In 2000, this looked really great, let me tell you. So I want you each to look at these things. So I want each person to claim an amino acid, and then I want you to look at how that amino acid moves. All right, does somebody want to take the northwest corner with amino acids 1 and 2? That's histidine and asparagine. Northwest corner. Let's see, the southwest corner is not very interesting. So let's look at the sort of east side right here. So why don't you take the northeast corner here, and you see how you have at the bottom here, you have sort of it's the whole south side. So why don't you take that one? And I want you to watch your region and see how things change as you're going from, here's protein A, and then protein G, so this corner, that corner, and then the south side down here. And I want you to watch, as the yellow things change, I want you to watch how your residues are rotating. And then I'll turn off the recording and we will discuss, and I will show it again.